did I know when I drank that beer, the party had just begun. When that yeast encountered a molecule of glucose, there was quite an attraction. Glucose knocked on yeast's door and went inside. After visiting for a minute, they decided they needed a spark to liven up the get-together. Glucose called up her favorite enzyme to grab ATP and bring him to the party. ATP arrived and got really hot inside, so he took off his phosphate and hung it on a glucose rack. Then ATP lost his energy and became party pooper, ADP. Glucose with the new phosphate transformed into glucose 6-phosphate. Now the party was really kicking off. Glucose 6-phosphate was needing one more Red Bull to make it through the next 2 seconds, so another enzyme brought a second ATP to the party. He also couldn't stand the heat and took off his phosphate and hung it on the opposite end of the glucose rack. This extra phosphate transformed glucose 6-phosphate instantly into fructose 1,6-bisphosphate. Where the party people at?
Officer enzyme arrived and broke the party up. Fructose 1,6-bisphosphate was out of hand and had to be split. Officer enzyme split one group into DHAP and the other into a group of G3P. Another officer enzyme decided DHAP was getting special treatment and was changed into another group of G3P.
So in this next step, we are in the energy conserving stage. At this point, the phosphates connect to the G3P and reduce the NAD plus and NADH, which means an electron is gained. In the next step, the phosphate from the 2,1,3-bisphosphoglyceric acid is removed by an enzyme to build 2-ATP. The remaining phosphates are moved to the middle carbons of the 2-phosphophenylpyruvic acid, which releases the water molecule from each substrate.
An additional enzyme breaks the bond between the phosphate groups in the second carbon and binds them to ADP, creating 2-ATP. The resulting molecules are 2-pyruvic acids. This process is done twice, resulting in a net gain of 2-ATP. Although 4-ATP were formed, 2-ATP were utilized in the beginning of glycolysis.